And today I'm going to talk a bit about the GNE Biobank that I'm helping establish in the lab, and also a gene editing strategy that I'm doing research on with some of the cells from the GNE Biobank. So first, about the biobank. So a biobank is a collection of biological samples that researchers can use for research purposes, including developing therapies such as the gene therapy that we're looking to develop for GNE myopathy. And it is a great research tool because it provides a centralized location to store and maintain cell lines. And it provides researchers not just in one lab, such as ours, but any researchers worldwide that want access to these cell lines, the ability to contact the NDF, and we can ship them these cell lines to anywhere. So as we're finding today, many of you have had the opportunity to donate samples to the biobank. So I'm going to talk about the process that occurs with these cells from the donation today to how they're used in research in the lab. So the first step in establishing this biobank is to collect the skin biopsy from both patients and unaffected family members. So this involves doing a simple skin biopsy, and the cells that we're looking for, fibroblast cells, are located in this layer, the dermal layer of the skin, which is divided into 3 layers. So the skin punch will go down to the second layer, and that is the fibroblasts or the skin cells that we're looking for. So after the skin biopsy has been collected today, the samples will be taken back to the lab, and they'll go through a process in which we extract the skin cells that we want, the fibroblasts, from the skin punch. So this involves a variety of steps, and I'll go through the first few steps right now. So the first thing is we receive the samples back at our lab at Yale, and this we will be doing tomorrow with any of the samples that we collect today. Then you remove the layers of the skin punch that we do not want, so the adipose tissue. You dissect out, and then you can process the tissue into smaller pieces that make it easier to access the fibroblasts. And then for the second half of this extraction protocol, we add an enzyme called collagenase, which digests the extracellular matrix around the cells. So it's basically cleaning the cells of the proteins that we do not want in the sample. Again, just trying to make sure that we're extracting just the skin cells that are useful for us to use in the lab from the whole biopsy. And then we can collect the cells, which you can see is a tiny pellet right here at the bottom of this tube. And then we can place them in a flask where they're allowed to grow out and expand into many more cells. So this growth of these fibroblasts or skin cells that we're looking for does take a while. So these are images taken from a microscope that can allow us to visualize these very small skin cells. So you can see at day 1, their cells are not attached to the plate. We can't really view them under the microscope. And then by day 7, you can start to see these fibroblast skin cells appearing in the culture. And over time, you can see many cells, many fibroblasts expanding from this tiny piece of the skin biopsy that we collected. And this allows us to grow from the tiny skin biopsy many, many cells. However, these cells cannot exist in culture forever as they are. So next we have to do a process called immortalization, and this allows us to maintain these cells in the lab growing in culture, so we don't have to go back to patients and ask for additional skin biopsies. So in this process, we introduce an immortalization agent into the cells and select for only those cells that have had the immortalization agent successfully introduced. We then confirm this process using a PCR reaction. We place the cells in tubes, and then we can store them in liquid nitrogen, which keeps them at a very cold temperature. And it is from this storage that we can then take these vials of preserved cells and ship them out to any other labs who may want to perform research on them. So in addition to looking at the skin cells, because GNE myopathy is primarily a muscle disease, we want to have some way of turning these skin cells into muscle cells. And the first step of that is to convert them from fibroblasts, the skin cells, into myoblasts, which are kind of a precursor to the muscle cells that we'll eventually want to study. So this is done again using a similar process to the immortalization, but instead of using an agent that immortalizes them, we're using an agent that helps convert them into myoblasts. And then when we do this process, we can see a morphological change in the cells. So we can see the shift from these fibroblast cells that are more spread out to the myoblasts, which are more elongated in structure. And then the last step would be then to convert these myoblast precursor muscle cells into the muscle cells themselves. So we induce the expression of the agent that does this, and it also expresses red. So when you look under a microscope, it's actually really cool. You can see that this agent is expressed because the cells will glow red under the microscope. So you can see here that we are able to successfully do this in lab. So all the cells that express this DS red have had the MyoD agent successfully transduced into them. And then we can also do an experiment looking at markers of muscle cells. Because as I mentioned before, it's really important for studying a disease like GNE myopathy that is primarily a muscle disease, that the cells that we're converting into muscle cells express the same properties as muscle cells. So here we're looking at myosin heavy chain, which is a marker of muscle cells. So here's in the top row, you can see what the expression looks like in a muscle biopsy, so directly from muscle cells. And in the bottom row, you can see what the expression of our converted muscle cells, so going from skin cells to muscle cells. And we see this same expression, indicating that we are successfully able to do this in the lab, which is really great because that means that when we're looking to establish cell lines to perform research on, we can take skin biopsies instead of muscle biopsies from patients that can give us a pretty accurate estimate of how the muscle cells behave. So the current GNE Biobank status is collected from a UCLA patient conference, and it currently is composed of 15 cell lines, 9 of which are patient samples, and 6 of which are family members, and they're all undergoing the process that I just talked about and are in various stages of that process, which takes between 5 to 8 weeks usually, depending on how fast the cells are growing. And we have already sent some of the cells who the process has been completed for to collaborators at Ohio State University and at Mount St. Joseph University, which again is just expanding the utility of this resource. We want it to be accessible to as many researchers at as many places as possible is the whole goal of this GNE Biobank. So that was a bit about how we create and establish a GNE Biobank, and then I'm also looking to talk about some of the science that we're doing with that biobank in the lab. So, and the first of which is a project looking at the determination of biomarkers for GNE. So one of the problems in the field is the lack of reliable and accessible biomarkers, so we're definitely looking to find more. And what a biomarker allows us to do is to look for a factor that differs between control healthy cells and then the GNE myopathy patient cells. So you can see here's an example where you see lower levels of intensity of this factor in the GNE myopathy cells compared to normal control cells. And this is useful because then when you're looking to develop therapies like gene therapy, you can see and measure, does this lower level in the GNE myopathy return back to normal levels when you apply the therapy or the drug or the treatment. So it's really important that we have these markers, so when we're looking to develop gene therapies and other therapies, we have a way of measuring the success of those therapies. So that is one of the projects that we're working on in the lab with this GNE Biobank. And then another project that I'm working on is CRISPR base editing to correct GNE myopathy mutations, specifically the M743T mutation. And what gene editing allows you to do is to go into the DNA, the genome of the cells, and hopefully correct a patient mutation. So it would take this mutation here, and through CRISPR base editing technology, convert it to the wild type healthy sequence. So you would have, in the lab, we would take some of the GNE Biobank cells that we've collected, apply the CRISPR base editing technology, and then you should have corrected patient cells that have in the GNE gene the correction from the mutant sequence to the wild type healthy sequence. So that is another one of the projects that we're looking to develop in the lab. And if anyone has more questions on that, feel free to ask me later. But with that, I would like to thank everyone in the LEC lab, especially Moncol, for his guidance, and a postdoc in the lab, Heather. I'd also like to thank the NDF for inviting me to come speak here, and then also a huge thank you to all the patients and family members that made this possible. Thank you.
So thank you so much. Can you guys hear me? Patrick? Oh, there it is. Thank you so much for that interesting presentation. Was that meaningful? Did you guys get a lot out of that? Does anyone have any follow-up questions or comments about what we're doing here at the LEC lab? Yes, we have a question over here.
Thanks for the presentation.
Yes.
You mentioned about biomarkers and how they are useful for gene editing or gene therapy trials.
Mm-hmm.
Are those biomarkers useful even for other trials, like for instance the MANAC trial?
Yes, so they're basically useful for any treatment trial that you're looking for. Basically, they're just useful because we want to find a way of measuring the success of the treatment. So it works not just for gene therapy, but for the enzyme replacement therapy as well, to look for an improvement in that biomarker once the treatment's applied.
And this is something we're going to be making available, right, Moncol, to other collaborators down the road, such as people on the man.
Any finding will probably be communicated. So yes, well, first of all, the cells will be made available to any researcher who wants to improve their biomarker development or want to discover new biomarkers, as we have already shared with other groups already. And also, and it's the hope that the scientific community does share any of their findings with everyone. So, so that industry and other people can actually use that as insight for what they're doing, and do it prior to publication also, because the scientists in the audience here know that the publication process is pretty slow and ancient. It takes about a year and a half. So, it's the hope to communicate it faster to everyone, not just wait for that publication. But that publication is a validation of the scientific community thinking that have sort of like validated the results were robust. So, even if we do share it early, it's something that people need to know that the scientific community hasn't reviewed it for robustness, and, and everything's done correctly and things like that. But definitely, like with everything, we'd like to communicate everything early.
Yeah. Thank you. Yes, we have a question over here. Let me get the mic to you so that we can hear the question on camera.
How you doing? This question might not be to your presentation, but you could probably help my understanding of it. As a caregiver for somebody who has, this type of muscular dystrophy, let me ask you a question. So, as I understand it, there are very many forms of muscular dystrophy, and
Yes.
we would. So, what we're working on to find a cure for this particular type of muscular dystrophy, in order for us to cure muscular dystrophy at all, we'd have to have several medications that we'd have to run through the FDA, yes, to cure muscular dystrophy. So, we're just looking at it, we're a foundation just looking at the cure for myopathy, correct?
Mm-hmm.
Right, GNE myopathy. So we're looking for a cure for just this type of muscular dystrophy. We're not really working on all cures for muscular dystrophy.
Would you like me to answer that, Moncol, or you want to take that? The hope is, and I'm going to have a scientist add to this, the hope is that with every bit of understanding that we have within these subcategories of muscular dystrophy, in this case GNE myopathy, they all have to be treated differently. There isn't a one-size-fits-all answer to all muscular dystrophy, but the hope is that as we learn more and more about GNE myopathy, that some of our findings can be applied across the board, across platforms to different types of dystrophies. Is that correct, doctors? Do I Can anyone add to that for the sake of this gentleman who asked that question?
Yeah, it's a great question, and like she was saying, each different specific muscle disease is going to be different in how it needs to be treated, but all of the work that we do for each specific disease is going to add to our understanding of that basic muscle biology that will help inform, hopefully, other therapies for other diseases as well.
Thank you. Did that answer your question?
Yeah,
Or you have a follow-up question?
yeah, one of the toughest things that I had to ask Kevin today, I asked him actually during lunch, was...
So you were saying that one of the toughest questions you've had to ask Kevin.
Yeah, one of the toughest questions I had to ask Kevin so far, and I asked him this during lunch because I needed to get in my mind a more clearer picture, you know? So the question I asked him was, if they come out with a cure before Kevin unfortunately passes on for his disease, it wouldn't help him because he's already too far gone, too far along in his progression of disease.
So I think people tend to conflate the word treatment with cure. So if there was a cure, then it would benefit him. But I think your question is, would there be a treatment within his lifetime? And I think, the jury's still out on that. This is how I answer the question, is that if everything that the scientists tell us at NIH and our scientists that are working on gene therapy, if they have their way, it will at the very least slow the progression of the disease so that at the very worst case scenario, he doesn't progress further. And Kevin seems like a very happy, productive human being to me, which is, this is a great way to be. So I think, does that answer your question? So the difference between treatment and a cure is permanent, and it's something we tend to shy away from because we really want to make people understand that a treatment is something that will address and tackle the issue, and the hope is that it slows the progression of the disease or halts it. And then there are some people in the field, and we're going to hear more about gene therapy this afternoon, where people believe that it can, in fact, also reverse the symptoms. So I'm going to leave that to the professionals to address later on. Perhaps if you guys could weave an answer to that in your presentations later on this afternoon. We have another question over here from Imad, who is always so vigilant with his questions.
I always try not to raise my hand, but my curiosity always gets the better of me. So I really found that CRISPR slide very, very interesting. And
Yes.
based on whatever I've read about CRISPR technology, it seems to be extremely promising, and just the design and structure of it, just based on my limited understanding of it, seem to be very, very promising, right? The only thing of concern to me in this case is the fact that GNE myopathy presents itself amongst different people in so many different types of mutations.
Mm-hmm.
I'm not sure what the cost effectiveness would be for such a type of treatment or cure, whatever it is. However, in that case, how expensive would a CRISPR treatment design would be, and would it be something that is it cheap enough that you can have like 4 different types of CRISPR technologies out there for GNE myopathy?
Yeah, so it is something that would be mutation specific for each construct that you're designing, but you do have the option and the ability to design gene editing constructs for a variety of different mutations. In terms of the final cost that would be to an eventual treatment, that would be more down the line for like pharmaceutical type of companies. So I'm not sure, but it's just definitely a therapy that we're also looking into in addition to gene replacement therapy, just because as of right now, we're exploring as many different avenues to find treatments for patients. And like for right now, looking at the mutations that are founder mutations, so we can try to expand that to as many patients as possible.
I'll just add to that. So
Let me bring you the mic. Let me know, because we want to capture this on film.
So I'll just add to Catherine's comment. So at the moment, CRISPR is not as mature of a technology as gene replacement. So what Katherine and others are doing in the lab is just showing proof of concept first and showing that, yes, we can target particular founder mutations. So we're targeting founder mutations because we can benefit the most people. So sometimes people have 2 of the founder mutations, sometimes they only have 1 out of 2, but if you only correct 1, that would be good enough for a recessive disease. So at the very least, what we can do in creating a proof of concept is create what I mentioned in my presentation, an isogenic cell line. So we can reduce variability in experiments because we have 2 cells, one with the mutation and one without, and no other genetic differences. So, and so that is critical in understanding more about the disease process itself in patient cells, and also development of biomarkers, where we've tried our best to reduce some of the variability that we know is contributing to variability in experiments.
Thank you. Raise your hand if you understood that. Thank you. Listen, guys, this is all tough stuff, and hopefully we can have this recorded. We're having it recorded so we can break it down, like if you're like me, with a dictionary in hand, and understand everything, and then call him up anyway and ask for questions. If there are no other questions for Katherine, I would like to thank her very much for her presentation.